Hi, everybody. I'd like to thank you for coming to today's Fish Health Seminar with UMaine's Aquatic Animal Health Lab. Today, I'm going to be presenting my research on establishing an Atlantic salmon primary gill cell culture for infectious salmon anemia virus ISAV-HPR0. To begin, I wanted to give a small background on cell culture. So cell culture is the process in which tissues are grown in an artificial environment. This was developed in the early to mid-1900s with Ross Harrison in 1907 growing the first nerve fiber to animal cell culture in the 1950s at an industrial scale. Animal cell culture is now a common tool used in laboratories for basic cell biology mechanisms, cancer research, toxicity testing, production of vaccines, and the growth of viruses for vaccine production, and most importantly for this research, viral research. So with that being said, let's talk a little bit about infectious salmon anemia, the virus that started this research. So infectious salmon anemia was first discovered in Norwegian aquaculture facilities in 1984. Since then, ISAV has been described in numerous countries, including the Faroe Islands, Scotland, United Kingdom, Canada, United States, and Chile. To continue on with ISAV, ISAV is an enveloped virus from the Orthomyxoviridae family of viruses, and it was characterized in its own genus, Ecivirus. Infectious salmon anemia causes the disease infectious salmon anemia that is seen in Atlantic salmon among other salmonid species. It is one of the few fish viruses listed reportable to the OIE, and detection of this disease usually leads to culling of pens and/or farms, leading to a lot of economic loss in numerous salmon farms depending on where you are. So something cool I wanted to share were these two photos. On the left, you can see this is a healthy culture of Atlantic salmon kidney cells. And on the right, it is a culture of Atlantic salmon kidney cells with ISAV infections. And it's really, really cool because I wanted to show you what it would look like on a microscopic scale. On a macroscopic scale, you can actually see tons of lesions on the inside and the outside of the fish. So there are two significant variants of ISAV. ISAV Delta, which is a high mortality variant, and a low mortality variant, ISAV HPR0. ISAV HPR0 is labeled as such due to the sixth segment that carries a full-length, highly polymorphic region as seen in the photo to the right. Culture of the non-delete HPR0 has yet to be achieved in a laboratory setting. However, there is some research investigating the ability of ISAV HPR0 transitioning or mutating into its high mortality, high virulent counterpart HPR delta. A slight issue, however, with the HPR0 strains is that it does not show any clinical signs associated with the virulent variants of the virus, leading to it being harder to detect in salmonid farms without proper testing. So this leads me to my next topic, and the question of all of this is why gill cells? So ISAV HPR0 has been detected in all major salmon producing countries, and research shown from Emelfot et al. 2016 says that the HPR variant has an affinity to epithelial gill cells of the gill tissue, opposite to the high mortality variant that is commonly found in endothelial gill tissue among other organ tissues. Culturing primary gill cells that could test positive for the HPR0 variant could aid in the understanding of HPR0 infections, possible mutations of the virus, or the role that HPR0 strains play in our understanding of ISAV and the infection route of ISAV. With all that being said, let's talk about the objectives that were laid out for this project. So the objectives we laid out for this project was to generate a reproducible method of obtaining primary Atlantic salmon gill cells, possible culture of ISAV HPR0 from Atlantic salmon that have tested positive for ISAV HPR0, and further investigation into infectious salmon anemia virus ISAV non-delete HPR0 strains. With all that being said, Atlantic salmon par were weighing 50 to 200 grams were maintained at the UMaine Aquatic Animal Health Laboratory to the right. The system was partial flow through at 14 plus or minus 2 degrees Celsius, and the fish were fed twice daily at a feed rate of 1.5% body weight per day. In order to collect the gills, Atlantic salmon par were euthanized with an excess of MS222 at about 250 milligrams per liter. Fish were bled using a 1-inch 25-gauge needle to remove the blood from the gills and the body. Once the gills appeared pale, the gills were removed using sterile dissecting tools, and the picture to the right is gills removed from a salmon after removing the blood, so this is what they would look like when they appear pale. As we know, gills are extra internal organs that can be full of contaminants. And in cell culture, contaminants are one of the biggest issues that we run into. So we did a small study using 3 disinfecting solutions, a 0.06% bleach solution, a 3% hydrogen peroxide, and a repeat wash of sterile DPPS, or Dubacle's phosphate buffered saline rinses. Contamination occurred in both the rinses of sterile PBS and 3% hydrogen peroxide. Furthermore, hydrogen peroxide had a strong reaction once the gills were placed into the solution. However, a 0.06% bleach solution allowed for the growth of the gill cells and the removal of contaminants. With all that being said, we decided on a 0.06% bleach rinse for about 30 seconds, following with 2 rinses of sterile PBS to remove excess bleach, blood clots, and possible contaminants. So what does the process of obtaining primary gill cells of Atlantic salmon look like? So it all begins with the euthanization of salmon parr and removing the gill arch and lamellae using sterile dissecting tools. The gills are then placed into a 0.06% bleach solution for 30 seconds. Once then, it is removed from that solution and placed into 30 mils of sterile DPBS twice to remove excess materials and contamination and blood clots from the gills. The gills are then placed into a sterile 100 millimeter Petri dish, and the lamellae are removed from the gill arches. The gill lamellae are then diced and placed into 4 mils of 0.05% trypsin and agitated for about 20 minutes at 175 rotations per minute. This is done twice before the gill material is put through a metal cell dissociation kit, and a cell scraper is used to push those cells through the sterile metal cell dissociation kit to further disaggregate the cells. Once that is complete, to neutralize the trypsin from further digesting the material, 1 ml of fetal bovine serum is added to solution, and the trypsin is neutralized. The gill material is then centrifuged at 3000 times G for 10 minutes, and then the gill material is then suspended in L15 plus 10% fetal bovine serum and gentamicin. This is all then placed into a 25 centimeter squared culture flask, and the media is replaced at 24, 48, and 72 hours post-culture. The media is then replaced every 3 days after until cells reach 100% confluency or begin to die. So what do the results from this look like? Overall, we have had success in achieving monolayers of primary gill cells in about 50 to 60% of our culture samples. We have observed fibroblastic and pavement cells in most of our cultures, and the cells continue to grow and divide until about 14 to 21 days, where they begin to vacuolize and die. Furthermore, we have not been able to successfully passage any of our gill cell samples. So I wanted to show some really cool pictures on your right, and this is what the cell culture flasks look like post-gill collection. And furthermore, I wanted to show various stages of confluency while culturing primary gill cells in our samples. So that first picture that you see in your upper left-hand corner is a gill lamellae that was placed into a 24-well plate. The cells are actually growing outside of the gill lamellae, which was something really, really cool that we saw. That second photo that you see in your upper right-hand corner is a cluster of gill cells 1 day after culture, so that second day of culture. So it's really cool. You see those cells dividing in that picture. In your lower left-hand corner, that is a photo of primary gill cells 3 days post-culture. So you see a lot of singular cells, but also small clusters in there. And finally, in the last photo in your lower right-hand corner is a full confluency of gill cells about 7 days post-culture. To further optimize our method, we decided to look into different sample tissue weights. We decided to test 2 grams, 3.5 grams, and 5.0 grams of tissue weight, and this is the lamellae tissue weight. Overall, our samples containing about 2 grams of tissue had the highest confluency in all of our samples. We attempted to subculture and pass these cells, but we were not successful once again in this. To emphasize that 2.0 grams of tissue weight, we attempted 8 more culture samples containing about 2.0 grams of diced gill lamellae. You can see the actual weights in that table to your left. There was no contamination in any of these flasks, showing that bleach rinse solution does work, and all the samples achieved confluency of about 70 to 100%. We were not able to subculture any of these samples once again. So the objectives that we have achieved so far is generating a reproducible method of obtaining primary Atlantic salmon gill cells. And we have achieved this at confluency in about 50 to 60% of our culture samples. However, the other two objectives of possible culture of ISAV HPR0 from positive HPR0 fish and further investigation into ISAV non-delete HPR0 strains is future work. To elaborate on a little bit more on that future work, our future directions for this research include testing gill tissue weight of less than 2.0 grams, successful passage and subculture of gill cells, possible culture of Atlantic salmon gill cells that test positive for ISAV HPR0, or the testing of salmon serum instead of fetal bovine serum, or a higher percent of fetal bovine serum in our culture media. So what are the key takeaways from this research? Infectious salmon anemia virus HPO0 is a low mortality variant of ISAV. It is found in all salmon-producing countries, and ISAV HPR0 has been shown to have an affinity for epithelial gill cells. Generating a method of obtaining primary gill cells from Atlantic salmon has been achieved with success with about 60 to 100% confluency in our samples. And then optimizing this method with gill cell culture using gill tissue weight that has been tested and confirmed to be useful with tissue weights of about 2.0 grams. Again, we're going to be testing lower tissue weights as well to see if that stays true. And finally, future research in culturing positive HPR0 infected gill cells could aid in a better understanding of the relationship between HPR0 and that high mortality HPR delta strains of ISAV, as well as the infection route of ISAV HPR0, which is something that hasn't been fully understood yet. With all that being said, I'd like to acknowledge my committee members, the UMaine Cooperative Extension Diagnostic Research Lab Aquatic Animal Health Lab members, USDA ARS Non-Assisted Cooperative Agreement, the Aquaculture Research Institute, my family and my partner. And if anyone has any advice or comments or any questions, you can email me at my email, or you can even message me at LinkedIn. Thank you so much for coming to my talk today and listening about my project.